The demand of commodities, fine chemicals, aroma compounds, proteins, or medicines such as antibiotics is increasing worldwide. However, conventional biotechnology production processes based on monocultures of microbial flora are reaching their limits. So how can new, more efficient production routes be created by scale-up that not only meet demand, but are also economically viable and sustainable? Roughly speaking, this is what the DFG Priority Program Interzel is about. We will be exploring this topic piece by piece with our podcast, Lab Whispers about bioengineering. Microorganisms are used to produce many of the products we use every day. Normally, many millions of cells are cultivated in laboratories and industry, but these results only give us an average value. Therefore, studies on individual cells can provide new insights. Today, we are discussing these topics with three researchers, Professor Alex Grünberger, Dr. Dietrich Kohlheyer, and Dr. Christian Dusny. Would you like to briefly introduce yourselves and tell us, for instance, how long you have been researching in the field of bioengineering?
Yes, thank you. So my name is Christian Dusny. I'm a bioprocess engineer by training, and I'm currently heading the working group Microscale Analysis and Engineering. And my specialty is actually the development of new analytical systems or technologies to quantify the performance and behavior of individual cells.
Thank you, Dr. Dusny. Dr. Kohlheyer, could you please introduce yourself?
My name is Dietrich Kohlheyer. I'm heading the research group Microscale Bioengineering at the Research Center in Jülich. I would call myself by training a microsystems engineer, and I have around 20 years of experience in the field of lab-on-a-chip and microfluidic technology.
Yes, thank you. And now we have Professor Grünberger.
Yeah, my name is Alexander Grünberger. I'm currently heading the group Microsystems in Bioprocess Engineering here at KIT. I have around 12 years of experience bridging the gap between bioprocess engineering and microfluidics.
I see. Thank you. And how did you all become involved in the overall program?
Interzel is this priority program of the DFG. And the interesting things about these priority programs is that you can join with several researchers. And the three of us applied for one joint project. And in the end, we got funding for our project. And this was a great opportunity for us because we are all engineers, and it is quite difficult to get funding for a project with three engineers in the field of life sciences.
That's wonderful. So tell me, what is the goal of your research project?
Yeah, the overall goal of our research project is to establish microfluidic technologies as we develop into the field of bioprocess engineering. And we are doing that by combining different microfluidic technologies and applied to the chosen examples to analyze the interactions of co-culture systems. And by that, we are not only learning from the co-culture and getting insights to control them, but also, I would say, get all the other researchers to be introduced into these microfluidic technologies.
A few years ago, I would say we were always making nice images, images of single cells, single bacteria. We could follow bacteria over time. We could monitor their growth. We could judge if they are healthy or not healthy. But if you're a process engineer, you want to know more. You want to know what is the productivity of the cell, how much product does it actually secrete, how much sugar do you have to throw into your cultivation system to get as much product as possible? But we couldn't measure that. And we were always, when we were meeting, we were always making fun. We have to move forward. We have to move beyond nice images. And actually, when we started Zimbal, that was always our goal, beyond nice images and quantify microbial performance. And talking about microbial performance, there are a couple of well-known microbial performance indicators, which a typical bioprocess engineer definitely wants to know. And so in Zimbal, also in the former Zimbal 1 project, we are focusing on quantifying cell performance and getting these performance indicators. And in Symbol, we can nicely distribute these performance indicators among the different research groups. So Alex, Christian, and myself, we all deliver specific performance indicators to get the full picture of the culture performance. And that is something very unique. There was no one before who could actually do that. And so I think we have been successful in moving beyond nice images so far.
Yes. And in your project Symbol, microfluidic single cell analysis is important. That sounds really exciting because cells are so small. How do you measure and model a single cell resolution?
Due to the size of the microorganisms, our most important instruments are optical instruments, so microscopes. So we can visualize the individual cell behavior. But the nice thing about this SPP is that we can also develop our methods further. So to measure cellular properties of biotechnological or process importance, like biomass or individual productivities of the cell, so that we can really see and understand the contribution of individual cells to the overall process output in the end.
Are you going to be able to transfer this research to an industrial scale?
It's always the question what you want to do with it. At the moment, I think we are doing mainly analytics. So we are developing tools to analyze microbes at an extremely small scale. So we are looking at the performance of individual cells or small cell communities. But if you're talking about cultivation devices, I think it is not our present goal to scale this up to large dimensions. There are better systems for that. But microfluidic systems currently are mainly used for analytical purposes. And if you want to use that in an industrial company, I think we are getting closer. There has been definitely tremendous progress in the last 10 years, in particular in the field of image analysis. There has been tremendous progress, for example, using AI-based image analysis methods, because you have to detect the cells, you have to count the cells, you have to track the cells over time. And developing software tools to do these tasks is quite a thing. And there has been a lot of progress. And for example, in my group, we have now very nice tools which certainly have the potential to go for let's say, commercial applications.
Like for instance, drug research or...
Yeah, for example, in the field of antibiotics, you could use these kind of devices to study the effect of certain antibiotics on the growth, on the health of cells, but also in the field of cell screening, strain screening, so you could look for the best performing strains. In the field of, let's say, pre-cultivations, you could do pre-cultivations at a very small scale, but with extreme high throughput.
I totally agree with Dietrich. So in the first instance, our methods enable us to acquire unique knowledge about cells, about the behavior, and we can use this insights to improve bioprocesses. This means we are not generating new strains or generating new production processes, but we rather use the new data to see how can we improve bioprocesses, how can we fine-tune it. And of course, these systems have also the ability to maybe in future serve as novel screening technologies. For example, screening process conditions, defined ones, which are not possible to perform with conventional devices.
So I see you're already working on the large obstacles and challenges that you're confronted with. Are there certain challenges that you'd like to talk about?
Yeah, working to develop and establish new technologies has always a lot of challenges. Of course, the technical ones, this includes that you develop something new and you have to optimize it to see if it fits to your workflow, if it fits to your experiment, if it fits or allows you to make the experiments which you would like to do. Yeah? The other thing is reproducibility. In bioprocess engineering, for us, it's really important that the technology not just works once, but works reproducible. We want to measure a behavior of a cell in a reliable way, and this is important. We have to optimize the technology that it's capable or possible to do that. But it's not just a technical challenge, it's also about training the students, the people. We are in a highly interdisciplinary field, and people have to communicate between microbiologists, bioprocess engineers, microfluidic people. And this is not easy. Often there is not the same language. Often the principal methodologies are different. And this is something which already we had to learn. We had long discussions for our proposal, how we express something, how we can realize something. And this is something we have to teach the students, the next generation, to bring the microfluidic concepts into the field of bioprocess engineering.
Well, it all sounds really great. I'd like to thank you all for being here today, Professor Grünberger, Dr. Kohlheyer, Dr. Dusny, and I wish you continued success in your research.
Thanks.
Thank you a lot.
Yeah, it was a pleasure to be here. Thank you very much.
Thank you.